to educator.com. Today we're going to be focusing on cellular communication. Multicellular organisms like plants and animals need to coordinate their functions among groups of cells, and they do so by various methods of intercellular communication. Unicellular organisms such as bacteria can also coordinate their functions. For example, they may form a biofilm, and that requires many bacteria working together. We're going to begin by discussing the extracellular matrix. In previous lectures, I reviewed the internal structure of the cell. Now we're going to talk about the structure outside the cell. So the extracellular matrix actually consists primarily of glycoproteins. So glycoproteins, as the name suggests, are proteins, and they have carbohydrate chains attached. In animal cells, the ECM contains primarily collagen fibers. So these are a type of glycoprotein. And the collagen fibers are embedded in another type of glycoprotein called proteoglycan. So looking at a cell with its nucleus and various organelles, and then there's an extracellular matrix consisting of proteoglycan and collagen embedded within that, collagen fibers embedded in this network. There's also another glycoprotein called fibronectin. And fibronectin provides the connection between the extracellular matrix and integrins. So integrins are proteins on the cell surface. So they're cell membrane proteins, and they serve as an intermediary between the cytoskeleton and the extracellular matrix. So here we have an integrin, and then fibronectin. Let's make that yellow. So fibronectin right here is providing this connection between the ECM and the integrin. Inside the cell, you'll recall, is a cytoskeleton composed of microtubules, microfilaments, intermediate filaments. So it provides structure and support and motility for the cell. And now you have this integrin connected to the fibronectin and the cytoskeleton and allowing for communication between those two areas. We're going to begin by talking about communication in plants. So intercellular communication in plants. In plants, you may recall that there are channels called plasmodesmata, and these provide a means for communication between the cells because plasmodesmata are channels that actually pass through the cell membrane, through the cell wall, primary and secondary cell walls, and the middle lamella. And the cytoplasm of one plant can communicate directly with the cytoplasm of another. So substances can be passed from one plant cell to another. Activities can be coordinated this way. So plasmodesmata are an important means of communication among plant cells. Now let's talk about cell-to-cell communication in animal cells. Cell junctions are connections between cells, and they allow for communication as well as coordination of activities among these cells. There are three types: desmosomes, tight junctions, and gap junctions. First, let's discuss desmosomes. Desmosomes connect adjacent cells through proteins connected to filaments. So each of the adjacent cells will have a desmosomal complex composed of proteins. And these two come together and connect. It's often described as being like rivets. So the cells are essentially riveted together. And intermediate filaments attach to these desmosomal proteins. These are especially common in epithelial cells. Epithelial cells are cells that line surfaces or body cavities such as the respiratory tract or the skin. The skin, for example, is exposed to stressors, wind and rain and sun, and so they need to be held together more tightly. There's actually blistering diseases. One is called pemphigus vulgaris, so it's a blistering disease caused by defects in the desmosomal proteins. if there's defects in these proteins and the cells aren't held together, the result can be blisters. The second type of junction is a tight junction. So tight junctions are also often found in epithelial cells, particularly in cavities such as the bladder that need to hold fluid, because a major function of tight junctions is to prevent leakage of fluids from between cells. Tight junctions hold the cells together right flush next to each other at what's called the apical end or apical surface of the cell. So it's a narrow band that seals, so the tight junction is right there, and it's a narrow band that seals these two cells together. So the apical surface, let's say you have a cavity such as the bladder. So if this is the bladder, fluid is within the bladder. The side of the cell that is next to the fluid is the apical surface. So if there's a whole ring of cells, within the apical surface would be the urine. And what this tight junction does is it prevents whatever fluid is within the cavity from leaking out between the cells, which is obviously a very important function. This surface that faces away, that is not in contact with the fluid, the opposite face, is called the basolateral surface. So again, this is a tight connection between the apical surfaces of cells, especially common in epithelial cells. The third type of junction is a gap junction. And we're going to talk about these in more detail when we talk about the neurological system, the nervous system. Gap junctions are actually similar to plasmodesmata in plant cells because these are channels that allow the passage of substances directly from the cytoplasm of one cell to the cytoplasm of the next cell. Channels that transport substances from one cell to another, directly from one cell to another, the cytoplasm of one cell to the cytoplasm of another. These often transport ions, and recall that since ions are charged, the transport of them would actually change the membrane potential of the cell. So in this way, cells can be coupled electrically and coordinated. And it's actually gap junctions that coordinate the contraction of the heart muscles. Neurons are coupled by synapses, and again, we're going to talk about these in more detail later on in the course. Okay, so we talked about various connections between the cells. Now we're going to talk about means of cell signaling. And cells communicate with each other through both electrical signals. So I talked about electrical coupling briefly. They also communicate through chemical signals. For example, substances that are released from one cell can bind to receptors on the target cell. Before we go into depth about that, let's talk about the two general types of signaling. Signaling can occur through direct contact. Signaling can also occur over distances. Plasmodesmata and gap junctions both allow direct communication, direct contact through direct communication, because substances are transported from the cytoplasm of one cell to the cytoplasm of the other. In plasmodesmata, the cytoplasm are directly communicating. So that's one form of direct contact. A second form of direct contact can be similar to this, but what can happen is one cell has a receptor and the other cell has plasma membrane proteins that can bind to this receptor. But they don't leave the cell and float over the receptor. Instead, they're actually cell membrane proteins, so the cells have to be in direct contact in order for binding to the receptor to occur. So direct Communication through direct contact can be either through plasmodesmata, it can be through gap junctions, or it can be mediated by receptors and cell membrane proteins that bind to the receptors. Communication over distances occurs via the release of a substance from one cell that then binds to the receptor on another cell. Sometimes the substance that's released is involved in just local signaling, local communication. So it's intended for recipients that are nearby cells, maybe to coordinate activities between a particular cell type in a tissue. However, communication can occur over much longer distances, and hormones allow that to occur. Hormones are released by cells in glands, such as the thyroid gland, the ovaries, the adrenal glands, and they're actually released into the bloodstream. Because they're released into the bloodstream, they can travel anywhere in the body to the target organs. So these cells are shown in close proximity, so this would be more local signaling. But a cell could actually be up in the thyroid releases its hormone, thyroxine, that can travel through the bloodstream and affect organs in different areas of the body. For example, thyroxine causes change in metabolism, it causes increase in temperature, heart rate, many blood pressure, so many functions throughout the body, and that's mediated by hormones and signaling over a distance. Local regulators only affect nearby cells. Hormones can affect distant target cells. We break cell signaling into 3 stages. The first stage is reception, the second is transduction, and the third is the response phase. And we're going to go into detail about each of these. So first, the reception phase. The reception phase is binding of a ligand to its receptor. This binding induces a conformational change in the receptor. Okay, so that induces a conformational change, a change in the shape of the receptor protein. Ligands can be neurotransmitters. They can be things like dopamine or norepinephrine. They can be hormones such as insulin. Most ligands are actually water-soluble. And the reason that most ligands are water-soluble is because the receptors are located on the cell membrane. So since the receptors are on the cell membrane, the ligands bind to those because they can't enter the cell and just bypass the receptor. Remember that a water-soluble molecule is hydrophilic. It's not likely to be able to cross the cell membrane. So again, most ligands are water-soluble. Most cannot cross the cell membrane. They bind to receptors on the surface of the cell. There's a major, very important exception. There are intracellular receptors, and hormones can actually cross the cell membrane and bind intracellularly. I'll talk about that again in a few minutes, but for right now, just know that the reception phase is the binding of the ligand to its receptor, and that induces a conformational change in the receptor. The second phase is transduction. When we talk about transduction, what we're saying is that the signal is being transduced or transmitted from the cell surface to the inside of the cell. So transmission of the signal from the cell surface. So norepinephrine or another substance binds to a receptor, and then that causes a change in the conformation of the receptor, which in turn can activate another protein, which can activate another protein, and that message is passed along into the cell until the final stage. So transduction is just the passing along of this signal. The signal is received at the cell surface, passed along to deep within the cell. So transmission of the signal from the cell surface to the interior of the cell. The final phase is the response. At last, after the binding and the transduction, the response occurs. So the signal that the cell received elicits some type of response. This could be, example would be transcription of a gene. It could be activation of an enzyme. And activation of an enzyme or transcription of a gene could result in a change in the metabolism of the cell. It could cause the cell to grow. It can even cause the cell to die. So the response depends on the particular signal that was received. So the signal is received, it's transduced inside the cell, and eventually a response occurs. To talk about the reception phase, we need to focus on the different types of receptors. There are three major types that you should be familiar with. G-protein coupled receptors, ion channels, and receptors with intrinsic enzyme activity, such as receptor tyrosine kinases. We're going to talk in this slide about G-protein coupled receptors. So again, three major types you should know. The first is G-protein coupled receptors. And these get their name from the fact that they closely work with a protein called G-protein. So in blue, this is G-protein. And the G-protein coupled receptor is shown on the surface of the cell. Another name for these receptors is 7-transmembrane domain receptors. The reason that they're called 7-transmembrane domain receptors is that their structure is such that they actually have, they're a single polypeptide that has 7 alpha helices embedded in the cell membrane. So that's where they get this other name that you might hear used. Okay, so here we have the signaling molecule. It's been released by another cell, perhaps, and it's here in the extracellular space. We have the receptor right here in purple and the G protein. What you need to know about G protein is the fact that it exists in two forms. If it's bound to GDP, it's inactive. If it's bound to GTP, it's active. GTP is a form of energy used by the cell. It's similar idea to ATP. So beginning here with stage 1. In the first step, the receptor is empty, it's unbound, and the signaling molecule is floating around. This G protein is going to be in its inactive form. So it's going to be bound to GDP. Bound to GDP, and it's inactive. And it's located nearby this receptor, the G protein coupled receptor. So the signaling molecule binds to its receptor. And remember that binding of a ligand, such as a signaling molecule, to its receptor, it's a similar idea with specificity between an enzyme and its substrate. When binding occurs, the receptor undergoes a conformational change. So this G-protein coupled receptor, the receptor right here, is in a certain form. Binding of the signal molecule causes a change in form that allows the G protein to bind. A conformational change in the receptor, let's say right here, that occurs with binding, allows this G protein to bind to the receptor. Binding causes the release of the GDP and from the G protein, and instead binding of GTP. So it's now, the G protein is now in its active form. So this was stage 1. This is stage 2. G protein binds to the receptor, releases GDP, and binds to GTP. It's now in its active form. Once the G protein is in its active form, it then is released from the receptor. The receptor's done its job. It lets go of the G protein. The G protein is active. Now, what is this green protein here? Well, this green protein is an enzyme, and it is the next step in the signal. The third stage is that the active G protein is released from the receptor and binds. So GTP bound to the G protein. The active G protein binds to the enzyme, and it in turn activates that enzyme. So once that enzyme's activated, it can activate another protein and so on, and then we get into the transduction step and eventually the response. G protein is also a GTPase, meaning that it can hydrolyze the GTP on itself. So it's going to go from GTP to GDP, putting it back into its inactive form and stopping the cycle. So although this is a very powerful cycle, it also can be short-lived. So again, going through the steps, the first step is binding of the signaling molecule floating around, empty receptor, receptor attaches. Attachment or binding of the receptor to its ligand results in a conformational change in the receptor. The conformational change allows G protein to bind. When G protein binds to the receptor, it releases GDP and binds GTP, which activates it. The active G protein is released from the receptor and binds to an enzyme. It activates that enzyme. That enzyme now can activate another enzyme, and so on in the signal transduction cascade. Okay, so the first type of cell membrane receptor is the G protein coupled receptor. The second type we're going to talk about is receptor tyrosine kinase, sometimes called RTK. And this has what's called intrinsic enzyme activity. So recall that the G protein coupled receptor underwent a conformational change. It bound to G protein and activated it, but the receptor itself was not an enzyme. Very different with RTK because receptor tyrosine kinases are actually enzymes. And kinases in general transfer phosphate groups. They actually transfer the terminal phosphate of an ATP. And if you're talking about a protein kinase, then they're going to transfer the phosphate from ATP to a protein. So they transfer the terminal phosphate group from ATP onto the hydroxyl group of a protein. Here, the name tells you what type, tyrosine kinase. So it's going to phosphorylate tyrosine. There can be serine kinases. Various types of kinases that specifically phosphorylate certain amino acids on protein. Okay, so this receptor is a tyrosine kinase. It has the ability to phosphorylate tyrosine. And in this case, it actually performs autophosphorylation. That means that it phosphorylates itself. It phosphorylates tyrosines on its own protein. This receptor exists in two forms. The first form is monomer. So you see there's a single structure, single receptor, and that is an inactive form. And you see that right now it is not bound to a ligand. Upon binding of the signaling molecule to the receptor, two of these monomers come together to form what's called a dimer. They undergo dimerization, and the dimer form is the active form. Once in their active form, then they can perform the phosphorylation. So the structure of this is that there is a helix embedded in the cell membrane, and there are multiple tyrosines on this protein. So these are unphosphorylated, and then once the RTK is in its dimer form, it's activated and it can phosphorylate itself. So it's going to add phosphate groups. and it's going to use ATP. So 4 ATPs will be converted to 4 ADPs to provide the phosphates. This phosphorylated form of RTK is recognized by proteins within the cell. So these particular proteins don't recognize the unphosphorylated form. But once it's phosphorylated, then certain intracellular proteins will bind to the receptor molecule. And different proteins can bind. There's several different ones that will bind. And each of these could start a different signaling cascade. So that might be one cascade. And then there may be a different cascade over here. And this is very important because this one receptor can trigger multiple responses by the cell. And this is very helpful to coordinate cell functions. For example, growth. For growth to occur, organelles need to be duplicated, more plasma membrane needs to occur, replication of DNA. So in order to coordinate multiple processes within the cell, that's made much easier by the fact that several cascades can be started just by the binding of a single signal molecule. So again, there are two forms. There's the inactive monomer form and the active dimer form. Binding of the ligand to the receptor tyrosine kinase receptor triggers dimerization. Once RTK is in its active form, it can phosphorylate itself, the tyrosine amino acids on itself, and that puts the protein into a form that's recognized by proteins within the cell. These proteins bind, those proteins are activated, and they will activate downstream proteins, again, starting that signal transduction cascade. And multiple cascades can be started by the various different proteins that bind. The third type of receptor that we're going to talk about are ligand-gated ion channels. These function differently than the other types that we discussed. These open upon binding to a specific molecule. And they're called gated channels because they open and they close much like a gate. So they exist in two forms. So here's the channel, and in its closed form, molecules cannot get in. In its open form like this, certain ions, particular ions such as calcium, can enter the cell. So these are called ligand-gated, meaning that it's a ligand that opens the gate. They're receptors, so they bind ligands. And in this case, when a signaling molecule comes along, let's say that this is the signaling molecule, and we will say that these are calcium ions. So the signaling molecule comes along, it binds. Binding is going to induce this gate to open. So the gate is now over here. It's no longer blocking the channel. Once this channel is opened, calcium ions can enter the cell. Eventually, this signaling molecule will disassociate from the receptor. So the signaling molecule is still bound here because the gate is open. So again, channel was closed, signaling molecule bound, induces a conformational change that opens up the gate. Calcium molecules can enter the cell, and then this is going to eventually dissociate. So it's going to float away, and now the gate is going to be closed again, and no more calcium ions can enter. So the signal will be shut off. Once calcium enters the cell, that can trigger a response from the cell or a cascade, similar to what we talked about earlier on. We're going to talk about these in detail when we discuss the nervous system, because the influx of calcium channels are important for the nervous system. They're important for contraction of muscles and transmitting signals throughout cells. The the receptors that I've been talking about so far were all located on the cell membrane. We talked about G protein coupled receptors, receptor tyrosine kinase, channels in the cell membrane, ion channels in the cell membrane. However, there are also intracellular receptors. With the receptors we talked about previously, the ligands were water soluble. They were hydrophilic, they can't easily enter the cell, so instead they would bind to receptors on the surface of the cell. They can't bypass the receptors. However, hydrophobic molecules, such as hormones, may have intracellular receptors. So here we have a receptor. So steroid hormones can enter the cell because they are hydrophobic. Similar idea to what we discussed earlier in that there is specificity. So here is a hormone. Recall that hormones are released from glands. They can travel through the bloodstream to distant target organs. Initially, they're located outside of the cell. They can cross the cell membrane and then bind to their receptor. This forms what's called a receptor-ligand complex. This receptor-ligand complex can travel into the nucleus of the cell because, again, it can cross cell membranes. Once it's inside the nucleus, it can act on DNA. It can act as a transcription factor and activate transcription of messenger RNA, or it can actually turn off transcription. So in this case, you notice that there's no intermediate step of transduction. The receptor binds to the hormone, and this complex directly causes the response. So rather than having binding to receptor tyrosine kinase and then activating another protein, which activates another protein, and so on until the response, here we just have binding of the hormone to the receptor, which directly acts as a transcription factor. Here it says intracellular receptors are located in the cytoplasm or in the nucleus. So what's shown here is an example of a receptor in the cytoplasm, binds to its molecule, signal molecule, enters the nucleus. However, in some cells, the receptor is located in the nucleus. So the receptor's in the nucleus, and the signal molecule will enter the cell, travel to the nucleus, and then bind there. And then this, again, receptor-ligand complex can go ahead and act as a transcription factor, and it's already in the nucleus, ready to do so. So a typical hormone that would act in this way is thyroid hormone. So thyroxine, the hormone released by the thyroid gland, binds to a receptor that's located in the nucleus of the cell. We talked about the first phase, the reception phase. We also started talking about signal transduction, and now we're going to continue on in more detail to investigate that phase of cell signaling. So the first phase was reception. The signaling molecule binds to the receptor. And we're talking mainly about receptors that are on the surface of the cell membrane. So it binds to the receptor undergoes a conformational change, and that change activates another protein, which activates another protein, and creates what's called a signal pathway, signal transduction pathway, or a cascade. Sometimes it's called a signal cascade because it's one activation causes the next, It's a cascade effect. These pathways often use what's called second messengers, such as cyclic AMP. These are small molecules that help to transmit the message from the surface of the cell to within the cell. So other second messengers could be calcium, could be IP3 and diacylglycerol, DAG, and we're going to talk about each of these. Over here is the structure of cyclic AMP, and this is an important second messenger, so we're going to cover this right now. The first messenger, so there's a second messenger. What's the first messenger? The first messenger is the signaling molecule. It brings the message to the cell from elsewhere in the body, binding of norepinephrine or internally binding of a hormone. So that's the first messenger. It brings a signal. The second messenger carries that signal through the cell, helps carry it through the cell until eventually the response is elicited. G protein coupled receptors use second messengers, so I'm going to use these as an example. Just briefly reviewing how G protein coupled receptors work. I'm showing a G protein coupled receptor here already bound to its signal molecule. Since it's bound to its signal molecule, it will undergo a conformational change that allows the nearby G protein to bind to it. When G protein binds to the G protein coupled receptor, it releases GDP and binds to GTP. That puts it in its active form. Recall that once the G protein is in its active form, it is actually released from the receptor and it activates another protein. Now we're going to talk about what that other protein is. Located near the G protein is an enzyme called adenylyl cyclase. And what adenylyl cyclase does is it catalyzes the production of cyclic AMP. So it converts ATP to cyclic AMP, shown here, which is a second messenger. So the binding of this active G protein to adenylyl cyclase activates it. The result is going to be the production of cyclic AMP. So levels of cyclic AMP in the cell are going to be greatly elevated. Lots more of this floating around in the cell. So again, activation of the G protein by binding to its receptor. Active G protein dissociates from the receptor and binds to the enzyme adenylyl cyclase. Adenylyl cyclase raises the level of cyclic AMP in the cell by catalyzing the transformation of ATP into cyclic AMP. Cyclic AMP can then, in turn, activate other molecules. So we have the cyclic AMP that could bind to another protein. For example, a protein kinase. Then this protein kinase might go and phosphorylate another protein, which is often a kinase. And once it phosphorylates that kinase, it might activate it. And then that protein will activate another protein. And sometimes you'll see these type of phosphorylation cascades where one protein kinase activates another, which activates another, and that continues on. Phosphodiesterase is an enzyme. Phosphodiesterase is an enzyme that catalyzes the reaction where cyclic AMP is changed to just AMP. So that will actually deactivate the cyclic AMP. It'll just become AMP, and that'll stop this cascade. So this cascade is powerful, again, but short-lived because the cyclic AMP level is going to quickly rise once adenylyl cyclase is activated, but then once phosphodiesterase converts cyclic AMP to AMP, then there's no longer enough cyclic AMP around to activate this phosphorylation cascade or whatever enzymes are downstream, and so the process stops. So again, the idea with the second messenger is that it is a small molecule, such as cyclic AMP, that helps to transmit the signal from the cell surface inside the cell, and it does that by activating a protein. Cyclic AMP can also be a second messenger for cascades involving receptor tyrosine kinase. Signal transduction pathways often involve more than one second messenger. So in the previous slide, I focused on cyclic AMP. Other second messengers, cyclic GMP. I also mentioned calcium ions, as well as IP3 and diacylglycerol. So let's focus on some of those. So calcium, IP3, and diacylglycerol, or DAG. Again, we're going to talk about G protein coupled receptor. Starting out at the step at which the receptor binds to the G protein and activates it. So here is a different enzyme this time. So instead of the active form of the G protein, it's going to dissociate. And now I've got active form of the G protein. Instead of binding to adenylyl cyclase, in this cycle, it's going to activate a different enzyme. And this enzyme is called phospholipase C. Phospholipase C is located right near the cell membrane, and the reason is because its job is to cleave phospholipids, a particular type of phospholipids, into DAG and IP3. So IP3 and DAG come from cleavage of phospholipids in the cell membrane. So phospholipase C cleaves a particular type of phospholipid called PIP2 into IP3 and DAG. So that's why it's located, again, form follows function, so I'd expect phospholipase C to be located near the G protein, which activates it, and also next to the cell membrane so it can cleave phospholipids within the cell membrane. So active G protein is going to go over and bind and activate phospholipase C. Phospholipase C is then going to create IP3 and DAG. IP3 then acts as a second messenger by going over to the endoplasmic reticulum and binding to a ligand-gated calcium channel. This is a ligand-gated calcium channel. Recall that in a ligand-gated calcium channel, the gate is closed until a particular molecule binds. In this case, the ligand that needs to bind is IP3. So the IP3 comes over here and binds. The result is release of calcium into the cytoplasm, greatly increasing the levels of calcium in the cell. Calcium then continues on and activates other enzymes. So it continues on with this signaling pathway. Meanwhile, DAG can activate a separate pathway. So as you see, it can be quite complex when multiple second messengers are involved. Again, the starting point was binding of the signaling molecule and activation or conformational change in the receptor that activates G protein. Active G protein activates the enzyme phospholipase C. Active phospholipase C catalyzes the transformation of phospholipids in the membrane into two second messengers. One is IP3, the other is DAG. DAG, so we had PIP2, and that it was cleaved, it's within the cell membrane, it's cleaved into these two. DAG can go off and activate other proteins. For example, maybe a protein kinase, and that could start a chain of reactions to create a response within the cell. Meanwhile, IP3 can go over and bind the ligand-gated calcium channels in the endoplasmic reticulum. That's going to release calcium, and that's going to go ahead and continue the response. So one thing to note here, when we talked about cyclic AMP, the level of cyclic AMP was increased by adenylyl cyclase catalyzing the reaction of ATP being converted into cyclic AMP. So it was actually the creation of cyclic AMP. Here, the second messenger, calcium, the level is increased, but it's not through creation of calcium. It's just through the release of stored calcium from the endoplasmic reticulum. So that's a different mechanism to increase the level of a second messenger. What is very important about signal transduction pathways is also that they can amplify a signal. So binding of one signal molecule can allows the receptor to activate this G protein. The G protein can activate phospholipase C. Phospholipase C can then create multiple molecules of IP3. So the binding of just the one signal molecule can activate enzymes that can catalyze the reactions many times, create many second messenger molecules. All of those IP3s can then go on and open a channel. All of the DAGs can go trigger a pathway. So at each step, the signal can be amplified. Okay, so it started out with binding of the receptor by a signal molecule. The message is transduced through the cell, finally reaches inside the cell, and you get the response. So the whole point of the binding was to trigger some sort of response by the cell. When we talked about intracellular receptors, that happen in a very direct fashion. The receptor is in the cytoplasm or maybe even in the nucleus. It's right there. It can act directly in the DNA as a transcription factor without all the intermediary steps. Even if there are intermediary steps, so the result is still a response by the cell. So what are the types of responses? I also mentioned increasing or decreasing transcription of a gene. So if the cell is going to grow and it needs to make certain proteins, then it can turn the genes for that protein on. Messenger RNA is transcribed. It's transported to the cytoplasm. It's translated into a protein, and there are increased levels of a protein. So the response can be increasing or decreasing transcription, which ultimately makes more of a protein, or you'll have less of a protein. However, there's a second way in which the cell can respond. It can act upon a protein that already exists. So sometimes the pathway doesn't cause more or less protein to be produced. Instead, it works on a protein that exists and makes it more or less active. That change in protein activity can result in changes in the metabolism of the cell, the shape of the cell, the growth of the cell. It can even result in cell death. So you should be familiar with apoptosis. This is known as programmed cell death, and it is one outcome of binding of certain signal molecules to a receptor. Programmed cell death has a very distinctive look. It's different than death of a cell just by outside destruction. In order for growth and renewal, apoptosis has to occur. First of all, of course, damaged cells need to be eliminated. So if a cell's been infected by a virus or the DNA is seriously damaged, that may trigger this signal for apoptosis to occur. Apoptosis is also a part of development. It's a part of embryological development. For example, cells are pruned back in the nervous system. They're also pruned back between the fingers and the toes so that those don't end up webbed when we're born. So apoptosis is an important part of an organism, and it can be triggered by binding of a signal molecule. It has a very distinctive appearance. What happens is the DNA fragments, the cell shrinks, and it forms these what are called blebs on its surface. You might hear this called cell suicide also. It's programmed cell death. So end results of binding of a signal molecule can be growth of the cell, increase in metabolism. It could be release of a certain substance by the cell, or it could even be the death of the cell. Example 1, describe the structure of tight junctions and gap junctions. Okay, cells with tight junctions are close together. They're flush next to each other. So they are, adjacent cells will be very close together. And they're held in place by proteins near the apical surface. Adjacent cells held close together by proteins, by a band of proteins near the apical surface. Again, the apical surface is the surface that is going to be near the interior of a cavity, or the lumen of a cavity. So the apical surface is going to be on this side, and the basolateral surface is going to be the other side of the cell, near apical surface. Gap junctions. So first, this is tight junctions. Gap junctions are connection or protein channels that allow the cells to pass substances directly from the cytoplasm of one cell to the cytoplasm of the other. So these are channels between proteins, between cells. Channels between cells. And they allow the transport of substances from the cytoplasm of one cell directly into the cytoplasm of the other. How do these differing structures reflect the functions of these two types of intercellular connections? Well, if we think about tight junctions, one function they have is to prevent the leakage of fluids. And this structure reflects that because since the cells on the apical, the side of the cell that is the apical side is going to be the side that is next to the liquid. So if there's liquid in here, you can prevent the leakage of fluids from between the cells by having them pressed next to each other. Otherwise, the fluid would just leak out, and then the cavity wouldn't really be very effective. Instead, with the tight junctions, the fluid is held into the space that it needs to be in. This also controls the passages of substances. Substances cannot just pass through here. The only substances that can get through are ones that the cell allows to cross. So that's tight junctions. Gap junctions have different functions. One function of gap junctions is electrical coupling. Electrical coupling is the coordination of charge between cells, and this occurs in the nervous system. It also allows the heart to beat in a coordinated manner. Electrical coupling occurs when ions pass from cell to cell. Since they're charged, then they can pass that electrical signal along. So ions pass from cell to cell. And this occurs through gap junctions. So the structure of channels allows for that coordination. Describe the three phases of cell signaling. The first phase is the reception phase. The second phase is transduction. And the third phase is response. In the reception phase, the signaling molecule binds to the receptor and induces a conformational change. In the receptor, which activates the receptor protein. So that is the reception phase. In the transduction phase, the signal is transmitted from the cell surface to the interior of the cell. This often is mediated by a cascade in which the receptor activates a protein, which in turn activates another protein, and on through the cell. And these proteins may be enzymes. Transduction is often mediated by small molecules called second messengers, such as cyclic AMP, calcium, IP3. The third phase is the response phase. The message is finally received by its final recipient. So a response could be enzyme is activated, an increase or decrease of transcription. It could be growth or death of the cell. It could be release of a substance. So the response phase is the cell reacting to that binding of a signal molecule. The signal has been received. The cell reacts accordingly. Why are ligands for intracellular receptors either hydrophobic or very small? Ligands for intracellular receptors, these type of ligands, must cross the cell membrane to reach their receptors. Their receptors are located either in the cytoplasm or the nucleus of the cell. Recall that Molecules that can cross the cell membrane are small, they're nonpolar, and they're not charged. Or they're polar, but they're very small. So in general, though, a small hydrophobic molecule will be best equipped to cross the cell membrane. Steroid hormones meet the criteria of easily crossing the cell membrane, so they can bind to the receptor within the cell. What is the cell's eventual response to the binding of a hormone to its intracellular receptor? What is the mechanism by which this occurs? So when a hormone binds to a receptor inside the cell, a hormone, actually a receptor, let's just say receptor ligand complex is formed. This is either already in the nucleus, if the receptor is in the nucleus, or it's in the cytoplasm and it travels to the nucleus. So I'm going to say travels to nucleus. It may already be in there. And activates or suppresses transcription. The effect will be an increase in the messenger RNA or decrease, and then eventually an increase or decrease in the level of a particular protein. Example 4. How does signal transduction amplify the response of a cell to the binding of a molecule to a receptor? One receptor can activate many proteins. So the receptor is bound by one signal molecule, but that receptor can activate many proteins. Each of those can activate many proteins, or if it's an enzyme, maybe act on many substrates. Therefore, at each step, the signal is amplified. You start out with just one binding molecule, but maybe the receptor activates 10 proteins. Each of those 10 proteins can activate 10 more, and so on. So it gets amplified at each step, which is very important to the cell's functioning and very efficient.